This is lecture 2.1 for Bio 182 on population genetics. In the previous lecture, we talked about the genetics of individuals, whether those individuals were homozygous dominant, heterozygous, or homozygous recessive, the different alleles, different inheritance patterns. Now we're going to look at patterns of genetics in entire populations. So we're taking a step out from individuals to populations. So a population is a group of individuals of the same species living in the same area at the same time. A population can be any group of individuals, and the scale of the area that we're interested in depends on the researcher and their question of interest. So it could be a population of grizzly bears in the entire state of Alaska, or it could be the population of grizzly bears in a specific national park. It just depends on what question that researcher is asking. But a population will always be a group of individuals that are of the same species that live in a predefined area at the same time. And a population is the smallest biological unit that can evolve. Individuals cannot evolve, populations evolve, and so this is the smallest unit of a biological organism that we can study for evolution. So in population genetics, we are analyzing gene pools. So the gene pool consists of all alleles of all individuals making up a population. And microevolution is the change of the frequency of alleles or genotypes in a gene pool. So when we're discussing population genetics, we are talking about how many of each allele there is in a gene pool for a population and how many genotypes there are for each possible genotype, homozygous dominant, heterozygous, and homozygous recessive, in that gene pool for that population. So in this example, we have a population of frogs. These frogs have a trait that is controlled by a single gene. The dominant allele is big B, and the recessive allele is little b. To measure the amount of microevolution that is occurring in this population, we need to calculate the 5 different variables at 2 separate intervals of time. The first 2 variables are related to the allele frequencies, and the other 3 variables are related to genotype frequencies. So for the first set, which are the allele frequencies, we have to calculate the frequency of the recessive allele, which is little b, and the frequency of the dominant allele, which is big B. We can also look at the frequencies of the different genotypes. So we have the frequency of the homozygous dominant or big B genotype, the frequency of the heterozygous or big B little b genotype, and the frequency of the homozygous recessive or little b genotype. So when we're talking about allele frequency, we're looking at, for the first one, for little b, for the recessive allele little b. So we can look at the population here and count the number of little b's in this population. And we then divide the number of little b's by the total number of alleles. So in this population, there are 13 frogs. So there are 26 total alleles because these individuals are diploid. So there are 15 little b alleles in this population, which we divide by the total number of alleles, which is 26, for a allele frequency of 0.58 or 58%. We can do the same thing for the dominant allele, which is big B, where we count up the number of big B alleles in this population and divide it by the total number of alleles, which is 26. So in this population, there are 11 copies of the big B allele, and there are 26 total alleles in the population for a frequency of 0.42 or 42% of the alleles in this population are the dominant allele. An important thing to notice is that the frequency of the recessive allele plus the frequency of the dominant allele are equal to 1. So the frequency of the recessive allele is 0.58 plus 0.42, which is the frequency of the dominant allele, equals 1. And they should always equal 1. The next variables that we can calculate the frequency of are the genotypes. So we can look at the frequencies of the homozygous, recessive, heterozygous, and homozygous dominant individuals. So in this case, the frequency of the homozygous recessive or little b genotypes is going to be 6 divided by the total number of individuals in the population, which is 13. It's now 13 instead of 26. We're not looking at individual alleles now, we're looking at individuals. And so in this case, we have 13 frogs, and so we're looking at their total genotypes and not their alleles. So in this case, we have 6 individuals that are little b or homozygous recessive divided by 13, which gives us a frequency of 0.46. The frequency of the heterozygous individuals, or big B, little b, is 3 divided by 13, which is a frequency of 0.23, or 23% of the population is heterozygous. The last one is the frequency of homozygous dominant individuals, where the frequency of big B individuals is 4 divided by 13, the total number of individuals in the population, and that equals 0.31 or 31%. So notice again that the frequency of the homozygous recessive, frequency of heterozygous, and frequency of homozygous dominant individuals equals 1. 0.46 plus 0.23 plus 0.31. Those values added together equals 1. So in population genetics, we talk about the Hardy-Weinberg principle or the Hardy-Weinberg equilibrium. And this states that genetic variation in a population will be constant in the absence of microevolution. So we're saying that without microevolution occurring, genetic variation will be the same from year to year. So anytime you sample the same population, the frequency of those alleles and genotypes will stay the same because there is no evolution occurring. We define microevolution as the change of the frequencies of alleles or genotypes in a gene pool. And the Hardy-Weinberg theorem, or the Hardy-Weinberg equilibrium, is a state where microevolution is not occurring, and therefore genetic variation is remaining constant. This is an extremely important concept because it represents a null hypothesis, which is what we expect if there is no evolution. We expect no change in the alleles or genotype frequencies in a population if evolution is not occurring. And it also provides the framework for quantifying or measuring evolutionary forces. If evolution is strong, then we would expect the alleles and genotype frequencies to change drastically over certain amounts of time. And if evolution is weaker, then that change will be a little bit slower and a little bit more gradual. So this Hardy-Weinberg equilibrium came from two scientists whose last names were Hardy and Weinberg. And again, the Hardy-Weinberg equilibrium is a state where there is no evolution occurring in a population. So if a population is in Hardy-Weinberg equilibrium and you know what the allele frequencies were the last time you observed the population, you can predict the expected genotype frequencies in the current population. So what does that mean? That means that if we have a population of frogs that we've been studying for years, and so say we went and sampled their genetics back in 2000, and now we're sampling them again in 2020. If that population is in Hardy-Weinberg equilibrium, meaning that evolution, microevolution, is not occurring, that the allele frequencies and genotype frequencies will be the same from that 2000 sampling time to the 2020 sampling time. So there is no microevolution happening, and therefore the frequencies of the alleles and the frequencies of the genotypes are not changing over time. There are essentially five equations that are related to this equilibrium, and they are also related to those frequencies of the alleles and the genotypes that we discussed a few slides ago. So the first one is that the frequency of the dominant allele is equal to P, the frequency of the recessive allele is equal to Q, and if you add up P and Q, or the frequency of the dominant allele and the frequency of the recessive allele, it equals 1. The second set of equations are that if you add up the frequency of the homozygous dominant genotype, which we call P squared, so think P was the homozygous dominant, or the dominant allele, and so we have big B, so it's 2 P's, so it's P squared. We have homozygous or heterozygous, which is 2PQ, and homozygous recessive, which is Q squared. And if we add up the frequencies of those genotypes, P squared plus 2PQ plus Q squared, they should equal 1. So we're saying with this that the genotypes of all individuals that are included in this population should equal 1, which makes sense if we're including all individuals that are in that population. So why is this the case, that this is how those alleles and genotypes are represented in these equations? So for Hardy-Weinberg, imagine that you have a population where individuals moved at random and mated at random. So the likelihood of any particular combination of alleles coming together to make an individual is proportional to the frequency of the occurrence of each allele. So in other words, if you overlay the allele frequencies over a Punnett square with a big A little a by big A little a or monohybrid cross, you can estimate the frequencies of each genotype, which are homozygous dominant, heterozygous, and homozygous recessive. So if we look at this Punnett square, we know that if we take two heterozygotes and cross them together, we have big A, which is, if we think of them in terms of P's and Q's from the previous slide, the dominant allele was represented by P, and so the probability of having a big A by big A individual occur from this cross is P times P, which is also P squared. The probability of having a big A, little a gamete result is can either be little a or big A little a. So it's either QP or PQ, Q times P or P times Q. There's two instances of that. So you have 2 PQ for the heterozygote combination. Then you have the homozygous recessive, which is little a by little a. Though the little a's in our Hardy-Weinberg equations are Q's, so the probability of getting a little a by little a individual is little a times little a, or Q times Q, which is Q squared. So this is where we get those parts of that equation where we have P squared plus 2PQ plus Q squared equals 1 from this hypothetical monohybrid cross between two heterozygous individuals. So as with most mathematical models, there are assumptions to Hardy-Weinberg. Those assumptions are that organisms are diploid. They only use sexual reproduction, not asexual reproduction. Generations do not overlap, so when a new generation is produced, the parental generation is deceased. Mating is random. Population size is infinitely large. Allele frequencies are equal in the sexes, and that there is no migration, gene flow, admixture, mutation, or selection. These assumptions all occur simultaneously, rarely if ever in nature, and so this leads to the realization, which is very profound, that microevolution is an action in most cases. So essentially we're saying that microevolution is occurring in any population at any given time. It may be slow or it may be fast, but microevolution is occurring in most populations at all times. So the classic example of population genetics comes from the study of population genetics and malaria incidence. And from this, we have this phenomenon that is called heterozygotic advantage. So a heterozygotic advantage is essentially just that there is an advantage of carrying a deleterious recessive allele. So a heterozygote has some sort of an advantage over individuals that are either homozygous dominant or homozygous recessive. And for this malaria example, that comes in the form of sickle cell disease. So sickle cell disease is a human disorder that is caused by mutations to a single base pair sequence. So when this mutation occurs, instead of the DNA coding for glutamine, it codes for valine. So normal sickle cell disease affects your hemoglobin, which is the cells in your blood that carry oxygen around your body. Normal hemoglobin is shaped like a squishy donut because of the structure of the proteins that make it. And so you have this hemoglobin that is inside of your red blood cells, and you imagine it to be like those hemoglobins filling up a beanbag chair. They're going to fill it up, and it's going to be sort of squishy and easily movable and easy to change the shape of. When that mutation causes the sickle cell mutated protein, the hemoglobin tends to clump together. And so instead of your beanbag chair, your red blood cell being filled with nice squishy beans, it's essentially filled with sticks or skewers. And therefore it changes the shape of that red blood cell. And so instead of that red blood cell being that nice, soft, squishy donut shape, it's now sort of in this hardened crescent shape. Normal red blood cells are able to squeeze and bounce through your blood vessels with no problem, and therefore they can very easily carry oxygen to different parts of your body. Red blood cells that are affected by sickle cell anemia and have that rigid crescent shape tend to get stuck in your blood vessels, and they then cause you to have blood clots and things like that, and oxygen does not easily reach parts of your body. So those are sort of the consequences of an individual having sickle cell anemia. So there are 2 videos on the next slides. So I want you to go back and watch those in the lecture slides that are posted online. One is just a few minutes, the other one is about 15 minutes, and it will explain more about how we have come to understand that heterozygote advantage in sickle cell anemia and how that relates to malaria.